Hello, and welcome to this series of instructional videos for the MassAI software. I'm Morten Rasmussen, and I'm the developer of MassAI. A few episodes ago, we did a search where we searched an MGF file against the entire human proteome. In the previous video, we made a limited search against one particular protein, one sequence, and that was calreticulin. Instead, we added a few variable modifications in the shape of cross-link dead ends, and we also added a cross-linking reagent, as in BS3. The video ended where we started the search, and now the search has finished. It's time to take a look at the results. As always, when a search has finished, MassAI opens automatically a couple of windows to display the results. It doesn't matter if you do a complex search or a defined search, the same windows will always open. Now the output is a little bit different in this case because if you look at the proteins identified in this sample, we will find one protein only, and that being calreticulin. This is no surprise because that's all we searched against. We did not search against any other proteins. So there's no real need to mess around with this window too much. We'll close it down and take a look at the more interesting results, which is where we find in the result list scan by scan. We're presented with a number of tabs, as always. We have the first tab, which displays the results that we have found for non-modified peptides. We'll just click one, and the annotated scan shows up, just like in the previous video, and we have the annotated sequence below. Some of you might have noticed that we have little colored dots in between the residues. Now, this all boils down to the mobile proton model, which predicts where we would expect fragmentation to happen. In this case, we expect to see fragmentation happening mostly between the lysines and also the proteins. Now, we see that the most intense fragment ion here is a Y6, and we count 1, 2, 3, 4, 5, 6. It's right here. Between the lysine, which is what we expect. If we look into the modified peptides, I'll just move this out of the way, we will find modifications in the terms of many dead-end crosslinks with the BS3, where the BS3 has reacted with the peptide and then attached itself and then reacted with water to form a dead-end crosslink. This is useful because it can tell us what residues are particularly prone to reacting with the crosslinker or accessible to the crosslinker. Next up, we'll show the tab for the cross-linked peptides. In the previous search where we didn't work with crosslinkers, this was blank. There was nothing there because we didn't include cross-linking. What we see here in the table on the left is all the possible crosslinks that calreticulin can form. There's plenty of them, as you can see. The ones in green are the high-scoring crosslinks. We'll just pick one, and all the scans in which this crosslink has been observed are displayed out here on the right. You can see that we have results both from the fusion, from the Q-exactives, and a few from the velos. We'll just pick, sorted by the score. There we go. I pick one, and below it we have the annotated peptide sequence. Now you will notice that this window has changed slightly from what we normally expect in the sense that we have two peptides instead of one. We have both cross-linked peptides in here, and the cross-link takes place between the residues that have been highlighted in red. So we have these two lysines cross-linking together. As always, we can go down the list, look at the... I'm going to do this a little bit smaller. Here we go. Go down the list and you'll see there is a consistency between the individual scans where we have observed the crosslinks. Now, normally this is a pretty poor scan, and normally we would probably discard this as a proper crosslink because the scan really is poor. What we do see down here, this is interesting, we see the reporter ions from the BS3 crosslinks. Next to this tab, we have a list of the highest scoring crosslinks. So we just get all the crosslinks with the best score first, and you can go down to examine all the crosslinks. Now, going through lists like these, where you have quite a lot of crosslinks, can become confusing really quickly. You have generally no idea of how does this relate to the protein? How does this relate to the structure of the protein? And it goes even more confusing. You have more than one protein in your sample that cross-linked between the proteins. MassAI comes with a number of different ways to visualize your crosslinks. I'm not going to cover this in this video. That's beyond the scope of this. And we close this down. What you'll notice up here, that's apart from the obvious tabs that we've been working with so far, we also have a top menu up here. And in this menu point called Specialized Searches, cross-linking has an entire section to itself with different ways to visualize crosslinks. We will save that for a different video. This series of instructional videos is always growing. And you have a say in what videos that we should create. So if you have an interest in cross-linking or any other features in MassAI and you would like to see a video on your particular subject, do feel free to leave a comment in the comment field below. Be sure to click the subscribe button to keep yourself updated with the videos that we create. And until the next video, happy searching.